Basically in the last session, as we have generated the filtered VCF, filtered FASTA file, so now we are going to perform the analysis, phylogenetic reconstruction using the Mega. So first of all, whenever we are performing the phylogenetic tree construction, basically first and foremost thing, like you need to align the sequence, because once you align, then you will be able to find out like how they are related with each other with the because it will show you the percentage identity, or it will show you the similarity or differences between the sequences. So here you have to just go with the align, say edit and build alignment, and then retrieve the sequence from file, because we are going to retrieve the sequences from our file. Okay, so you just have to click on OK. Then you need to select a folder where you're having your input file. So you have to go with the phylogenetic tree folder and select the filtered OTU file and open it. So here, if you will see, once you open all the alignment, all the 92 sequences, so right now this alignment is not selected. Those sequences are not selected. So here you can see only this first base is selected. So if you want to select
Ma'am,
all,
can you repeat?
Excuse
me.
Just a minute, let me complete. So if you
Okay.
want to select all, you have to select the control A, and then you have to go with the alignment. So for the alignment, what it is asking, like it is asking that you want to perform it with the cluster W or a muscle. So we are going with the cluster W because it's a multiple sequence alignment tool. So we are going with that. And yeah, so what you have to do, sir, you have to go with the alignment option, retrieve sequence from file. You have to select the FASTA file, and then uh, you have to just simply select all the sequence by the control A and go with the
Ma'am, I need to
alignment
click
option.
on the click on the file icon.
Alignment.
I have align icon. Where is the I can find alignment?
Yeah, you have to go with the align option.
Okay. So after align, I need to click open
Retrieve
the saved
sequence.
align. I cannot see the retrieve sequence. Edit view build align tree.
Here you
I
have the
cannot.
align option, edit and
Yes,
build
ma'am.
alignment, retrieve sequence from file.
Okay, wait. Let me come here so that I will.
Sir, what I will suggest you for today, because this is our
Uh-huh.
last session, so just watch it carefully for today, what I'm performing, and just perform it by seeing the recording completely. Because there are so many steps and so many parts. So, is it better that if you will just watch it completely, all the steps, and then perform?
Okay.
The choice is
Okay.
yours. You can tell me if you want, I can wait, but it will take a very long time. And
No, that's fine. I can go with the what you are suggesting. No problem.
yeah, so that like you can completely focus, right? I'll go it slowly then.
Yeah.
Yeah. And whenever, because this is our last session for the metagenomics, from tomorrow I am not available. So basically, I have to finish it for today. But still, if you have any questions, we can arrange one doubt session on Saturday. Okay?
Okay.
Yeah. So as we are going with the alignment option, we are going with the edit and build alignment. And as we have to retrieve the sequence from file, so we are going with the retrieve sequence from file and say OK. So I'm selecting here the folder where we are having the uh, phylogenetic tree filtered OTU we have to select. And here after that, you can see only the one base is selected, right? We have to select all the sequence. So for that, we are doing the control A, which is going to select all the sequences. And then here we are going with the alignment option. So for the alignment, there are the tools which we are having. One is a cluster W, another is a muscle. So cluster W actually is a uh, more appropriate and like a advanced tool which we are using right now, which is updated one. So better that you can go with the cluster W. And here, after that, it is asking, like it is performing the pairwise alignment and the multiple sequence alignment. But in the pairwise sequence alignment and the multiple sequence, it is showing you the options gap opening penalty and the extension penalty. So, what is the meaning of opening and extension penalty? Any guess?
No.
Okay, so gap opening penalty is what? Like, this is the cost of introducing a new gap in the alignment. So, higher the gap opening penalty makes it less likely for gaps to be introduced. Because obviously, if you want to find out the similarity or you are performing the alignment, we are not preferring the gaps over there. So, if you will give the high gap opening penalty, there are less chances that you will introduce a gap inside it. So, that's why the default value actually is a 15 for the gap opening penalty. Then another is gap extension penalty. Like this is a cost of extending an existing gap by one position. So lower the gap extension penalty makes it easier to extend an existing gap. So higher we want the gap opening and lesser we want the gap extension. So what are the default values in the mega? So basically, this mega it provides a default gap penalties, which is generally suitable for wide range of alignments, and these are definitely based. Like these are the default based on the empirical data, which is a common in use. So what is the default value? It's 15 and 6. But means like if you will just give the example, like it takes one example. If like if you're giving the gap opening penalty and extension, you can put 15 and 16, perform the alignment and evaluate. If you're not getting results whatever you are expecting, you can lower the gap opening penalty and lower the gap extension penalty to the 10 and 3 and perform the alignment, and again you can compute the results. But still, if your alignment is still not optimal, you can try with the 40, 12 and 4 as the cutoff. So this is how you can adjust the cutoff over here. Okay. So this is what basically we need to give these options, or you can change if you want. And here it is performing the alignment pairwise and multiple. Yeah. So here if you will see that the alignment is done now. So once this alignment is done, we need to export this alignment in the mega format. So here we are going with the data, then export the alignment. Okay, but in which format we are going to extract it? In a mega format. So you just have to select a proper folder where you want to save this and save. So here you can give your input title mega. You can give whatever you want. The choice is completely yours. So protein coding nucleotide data. So we don't have protein coding nucleotide data. Our data is a like a normal sequences. So here you just have to say no. Now once the alignment, once the mega format is done. So after that here once we saved the input in a mega format, now we are going to construct the phylogenetic tree. So how we are going to construct the phylogenetic tree? Here we are having the options for the phylogeny here. So in the phylogeny, basically we are having a total five algorithms or a five uh, options by which we can construct the phylogenetic tree. But which we can use? What is the meaning of each and every? That I will just explain you theoretically. So the first method we are having, that is a maximum likelihood. So what this maximum likelihood it is doing, it is going to evaluate the different possible trees, and then it is going to choose the one that maximize the likelihood for likelihood of observing the given sequence data under a specific model of evolution. Means what it is doing, it produces the highly accurate tree for the complex modules as well, but actually it is a computationally intensive and it's a complex algorithm. Another we are having, that is a never joining algorithm, NJ. So never joining is what, it is a distance based method that is going to construct a tree by iteratively finding the it's like what it is going to iteratively finding out the pairs of OTUs, and then it is going to be minimize the total branch length at each step. It is fast and efficient for the large scale data as well, but here you have to compromise with the accuracy then, because it is a less accurate. Then third we are having, that is a minimum evolution. So minimum evolution, what it is doing, it is finding the tree with the shortest total branch length, which is aiming to minimize the overall evolutionary distance. It is aims to find a tree with the shortest total branch length, but it can be less accurate again and sensitive to errors in a distance estimation. Then other is UPGMA, which is unweighted pair group with arithmetic mean. That is a UPGMA. So basically, this is a hierarchical clustering method, which is assuming that it is at a constant rate of evolution, uh, and it is going to be build a tree by clustering the pairs of taxas. You can say which is based on the average distances. Means it is simple and fast method, which is assuming a constant rate of evolution. But again, here the accuracy is less if the assumption of your evolutionary rate is violated. Like if your evolutionary rate is violated, that time it can be less accurate. And the last one we are having, that is a maximum parsimony. So it is producing a this is going to be identifying the tree topology, basically, which is required for fewest evolutionary changes, or you can say mutations to explain the observed sequences. It seeks the tree with the fewest evolutionary changes, but it can be a misleading when you have the evolutionary rate which is widely vary among the different branches. So this is what a total five algorithms we are having. But now again, how we can make a decision, which one we can use. So decision making is simple. If you are seeking for the accuracy, if you want a more accurate results, go with the maximum likelihood, the first one, because it is generally considered for the best choice if your computational resources permit as a incorporates detailed model of a sequences. But if you are seeking for the speed and efficiency, so go with the neighbor joining. And if you want for a specific data sets, go with the UPGMA. Means in a conclusion, what I want to say, the best method is depend upon the balancing the need of accuracy, computational feasibility, and the specific characteristics of our data sets. Because here multiple methods you can perform rather than that. Like still if you are confused, if you are not sure, you can you can perform with the multiple methods and then compare their robustness, their reliability for the phylogenetic tree. Okay, is it clear? Hello. Hello, is it clear to everyone?
Yeah.
Okay. What about others? Okay, fine. So now we are going with the maximum likelihood method. And then here we have to simply give the input of our filtered OTO mega file, MEG file. Yeah. Now here in your system, what it will show you like this test of phylogeny, none. But here we are going to use the bootstrap method. Okay, instead of this, you have to use a bootstrap method because it is a technique which we can use to estimate the reliability or the robustness of a tree. Means, what is the purpose of this bootstrap? It is to access the confidence level, you can say, or to support a various branches in the phylogenetic tree. Means it helps us to determine like how strongly the OTUs of taxas, or you can say a species or a sequences, is supported by the data. So if you will give higher number, you will get a better confidence. Like minimum, you have to give 100. I'm repeating this again. At least you have to give 100. But as we have a less time, because 100 will take a lot of time, so I'm giving a 10 replications. But I'm again repeating it. Just focus that minimum number you have to give 100. Maximum you can give as per your choice, that how much confidence you require. Okay? The rest of the things like a number of threads. So number of threads also, as per your system, you can mention and then click on OK. Any doubt here?
No, ma'am.
So
Okay.
maximum you said we can choose for
Hmm.
the confidence
Yes,
level, but
yes. But hello, uh, you're not audible, ma'am. Am I audible?
yeah, yeah, you are.
Oh, okay, okay. But can you just repeat your sentence? You can see one minute. On the downside, replicate like 5 out of 10 is done, 6 out of 10 is done. So it is completing the process now. Yeah, ma'am.
Okay.
Can you just repeat your sentence, last line?
Saying like for bootstrapping, you said like we should go for 100.
Minimum.
Yeah. And
Yeah.
what could be the maximum range? Like, because
Maximum
the
you can
more
give.
confidence level, it's better for the results, right?
Yeah, right. So there is a no access limit, I guess. But uh, generally, people are giving 500, 1,000. I'm seeing
Okay.
that. Yeah, but
Okay.
again, the choice is yours. But minimum, at least you have to give that number, 100 one.
Okay.
Oh,
Thank you.
okay. Okay, so here this is what our original tree it is. And here this is what our bootstrap tree. Okay. So bootstrap, sorry, original tree here, you can just simply see first of all the branch length options are given over here if you want to change. See here. Branch length is what? So basically, this is the measure of evolutionary distance. Means it is expressed in the terms of number of substitution per site, like nucleotide or amino acid changes that have occurred in the branches. So how we can measure this? So basically, if the unit of the branch length is typically substitution per site, so this unit is reflecting us the average number of changes, or you can say the mutations which is accumulated as per the site. Means for a nucleotide, if you are having like a branch length, it is measured in the number of nucleotide substitution per site, like point 0.1, which is zero point 0.1 substitution per site. For a protein, branch length are similarly measured in the number of amino acid substitution per site. Okay, so this is a branch length. If you want to visualize it, just select it. Otherwise, that's fine. Next, we are having that is a frequency info here, statistics, frequency or info. So basically, this frequency info, it will tell you, you can see it, that here it is showing you this frequency, it is showing you the bootstrap support values. These are the percentage basically, like 10%, 20%, 30%. So like, these values are indicating the percentage of bootstrap replicates in which the specific branch or a clade appear. Means if you are getting a 10%, so it will support what 10% is replicated, 20 is replicated. So this is what a statistical info. If you want to increase the font size for it, you can increase. You can place it above branch length, below branch length. So you can just edit as per the choice. After that, we are having the distance scale. So basically, this distance scale is also here. One second. Actually, this distance scale is we are having downside if it is present. So basically, if you are having a distance length, so it is going to show you the measurement of evolutionary divergence, or you can say a genetic distance between the sequences or a taxa or a species. Means, just take an example. If you have a distance scale 1.01 in the molecular phylogenetics, so it is indicating the moderate level of genetic divergence with the average of 1.01 substitution per site. Then here in the downside, you are having the ancestors as well. Like this ancestor is going to be refer the ancestral sequence, or you can say a node in the phylogenetic tree. Other options also we are having if you want to go with the subtree options, or a view if you want to see some different types, like root tree, or if you want to, sorry. Yeah, if you want to see some rectangle, straight or curved, so you can also select as per the choice. You can go with the circular also if you want. So this is how the editing also you can perform with the original, and here you are having the bootstrap tree, which is showing you the bootstrap replication. But one thing if you will observe here, still we are having this OTU IDs over here. We don't have the genus name, what it is representing. So we can't understand what exactly the organism is, right? So what we
Yes.
have to do, we have to edit this image, and for this editing, we are using one uh, online software which is ITOL, Interactive Tree of Life. So in this ITOL, if you will just go with this upload option, so here you have to upload your file, but this ITOL is accepting the input in a NAVIC format. So what you have to do is you have to export this result, go with the file, and export the current tree in the NAVIC format. If you want to display the bootstrap value, just select it and say OK. So here it will give you this NAVIC format. Save it. First of all, file, save as. You can select a folder where you want to save. Navic file, save it. Now, if you will go with the ITOL here, in this ITOL, you need to be upload your Navic file format. But one more thing again, how the ITOL will also know means like if I will just paste it this Navic format, Control A, copy, and here if I will just go with the Navic format and upload it, you can see how the ITOL will also know what is the file, what is the name of this OTUs, right? So in the ITOL, we have to give the annotations. Means what you have to do, you just have to download one file from the ITOL, and in that file you need to submit the ID, OTU ID, and what genus name it is representing. So for this, you have to go with the ITOL help. And in this ITOL help, if you will just go down, yeah. So if you will just go with this annotation templates file header, so in this you need to download the zip file which is given. Okay. Yeah. So once we downloaded the zip file, after that you can just copy this or cut this and put it into the phylogenetic tree folder. Then extract it. So right click and say extract here. So once, oh sorry. Okay. I think extracted many files here only. Just a minute. Okay, I will delete rest of the files. What we can do is we can create here one folder by the name of ITOL first. And inside that, then I will put this file. And then I'm just going to extract it now. Yeah. So once you extracted this file, after that, in this, you are having one file labels template. I will just copy this label templates for the better visualization and I will paste it into the phylogenetic tree. And once you'll open this file, downside if you will see, you have to paste your input like your OTU ID and the genus name, what it is representing. But how, in which format you have to paste? This is your ID, comma, your name, which means OTU ID, comma, name, genus name. So whatever the input file we are having, which we have prepared yesterday, taxonomy file. Yeah. So here, if you will see, we are having all this information in two columns, ID in a column and genus name in a B column. So what we can do is we can use one concatenate function in the Excel. So give the equals to sign and then we can use this concatenate. So what we want to concatenate like this value, comma, by what we want to concatenate by the comma. So in the double quote, I'm providing the comma. And after that, again, by the comma separated, this is what a second value means. A1, like the OTU ID, comma, in the double quote, again, you have to provide the comma and again this second name and then enter. So you can see This is what the OTU ID we are having with the genus name comma separated. Autofill it, and once you autofill it, you have to remove the formula. How? Copy, right click, paste to special, and here you have to select this values. So once you selected this values, after that you have to copy it carefully. Select the first value and control shift down. Copy, and then in this labels Excel template you have to paste it. Save this now. So once you save this file, now you have to go with the ITOL where you have uploaded the file. And in this, if you will see in this data set option, it is showing you to upload annotation file. So go with the upload annotation file, and then you have to select the labels Excel template and click on OK. So you can see it changed now. See, instead of OTUID, you are having the name, genus names over here. You can arrange, you can go with this basics and arrange this in a circular format so that you can visualize it properly. And then you can edit as per your choice. If you want to invert a tree, you want to change a font style, if you want to change the like a color. So all those you can just change as per your requirement. And then if you want to save it, go with the export. You can select the format like PDF you want, PNG you want, in which format you want, you can select and export it.
Ma'am, how to add bootstrap value in this tree?
Bootstrap value, okay, there will be an option, I guess so. Yeah, here. You can see here it is how added, okay?
Okay.
Symbols, with the help of symbols. Okay. I think numbers also it is added. We just need to increase the font. See, you can go with the text option over here. Okay.
Okay, ma'am.
Otherwise, if you want to display with the symbols or with the colors, you can just display. Okay. So here our targeted metagenomics is completed. You can just perform this once, and still if you have some doubts, we can arrange one doubt session on Saturday so that whatever your doubts, you can just first of all forward in the WhatsApp group. Just send it in the group instead of sending personally to anyone so that I can also check in the group, right? And then we will have one doubt session if you want.
Okay. Ma'am, how
Yeah.
to make the bar plots for this data?
Right. Bar plot is left, right. Okay. So here, actually, the command is given in the script only. That one command is left, I guess. If you will go with the script, so basically this last command is given for the bar plot generation. See? So Quime tags a bar plot. Then we are providing the input table.qza, then taxonomy, then metadata file, and the visualization. I will just
Mhm.
open the folder from the metagenomics. Then I just need to activate the Quime because this is a Quime under the Konda environment, Koyam environment. And this last one command we are running. Yeah, yeah.
Ma'am, apart from the workshop, I have a question.
Uh-huh.
So I contacted to Samiti ma'am and I have a doubts regarding one code. So she messaged me that I can directly message you and ask for the tomorrow's time. So I'm going to just message you. Please let me know what time you have so I can clear my doubts before joining the next course.
Sure, no problem. Can we meet at 5 p.m. then?
Okay, sure. I will
Yeah,
text you now. Thank you.
sure. Okay. Yeah, done. So here it is generated the QZV file. So basically, we have to go again with the Quime tools view. And here we have to drag this. Yeah, taxa bar plot, QZV. Okay. And here you can see this is what the bar plot we can visualize. You can increase the bar width. And this is what for kingdom bacteria, like a kingdom it is showing. If you want for kingdom, like all the 7 levels, you can select it. And here it is just displaying the bar plot, right?
Yes.
Is it?
Um,
Yeah. Okay, so here all the metagenomics part is done.
Okay. Thank you so much for
Oh,
nicely explanation.
yeah, you're welcome.
So could you please upload the recording in the, how
Yeah,
do we
I will
access it?
upload it by night, but the link you will receive by tomorrow only. Okay.
Okay. So it will be on your site.
No, actually, uploading I'm doing, but the link which is shared by the HR team, so
Okay.
they will join by tomorrow because our office timing is 9 p.m. till. It will
Okay.
take little
Understood.
bit time to.
Thank you.
Okay, so that's it for the today. If you have any doubt, please let me know. You can just put in the group and we'll solve it. Okay, so please just perform all the things which is done by today, and yeah, you will be able to perform.
Okay, ma'am. Thank you.
Okay,
Thank you.
yeah, you're welcome.